Hi, everyone. So we have our last lecture today, and I'm so excited we have Jamie Tyler. So as we talked about in our very first lecture, and many people have mentioned, Jamie was really, did a lot of early work in this area and kind of was sort of brought it back from the dead in a sense. And so he's been a real leader in the field in many ways. So I'm so excited to have him. I'm just gonna turn it over to him. Go ahead, Jamie.
All right, thanks, Kim. Thanks for inviting me to lecture today, and thanks, Keith, for being here to help facilitate our discussions. I'm excited to talk to you all. I'm always excited when I get to give a lecture at Stanford. I feel like I'm smart. The title of my talk is Neurofuss from Traw to Crest and Beyond, and I hope that the title becomes kind of clear as we go throughout the talk. We'll talk about some of the hardships as well as some of the fun times. So I'm also going to talk about my financial disclosure and try to explain to you kind of how we've approached the research and development. So I'm a co-founder of a company. This is my fourth company that I've co-founded. I'm an inventor on over 20 issued US patents and multiple international patents for various methods of neuromodulation. I have additional IP on psychedelic use and methods of administration of psychedelics. And a company in Toronto, Canada, that we help Diamond Therapeutics. And so this is all consolidated under IST as a private company. It's been really, I think, because I'm talking to you at Stanford, right? And I always think Stanford is kind of like, when you think about academic entrepreneurship, you think about the great people who have come from there, right? And so a lot of people try to model Stanford and the culture that they've created in that area in the Valley. And I think from my perspective, I've been really fortunate in that most of my research has been funded through private financing, private funding. We've raised collectively over $40 million, and I'd say probably a third of that has gone to research and development, helping push the field forward and across different gaps in ways that I think a lot of people won't ever understand. But it's been really it's been rewarding to be able to do this, and so I would encourage any of you that can think out of the box and can balance multiple things in your lives to think about entrepreneurial paths in ultrasound, especially because I tell all my students at ASU and biomedical engineering that ultrasound is like one of the most dynamic, hottest fields in all of engineering right now, I think. So I became interested in using ultrasound when I was a graduate student. This was the first paper that my PhD advisor gave me when I was studying my PhD. I was a synaptic physiologist. And Francis Crick, when he was at the Salk Institute, wrote this paper, Do Dendritic Spines Twitch? It was really a hypothesis that dendritic spines were contractile functional units in the nervous system, and they expanded and contracted in dependent on synaptic activity or synaptic inputs. And this was in 1982, right? And so that was a very interesting idea when it was combined with the kind of the first observations that dendritic spines are motile structures and mobile, that there's morphological plasticity in, kind of like across very small or short time scales. And so this was the first paper that I was exposed to in Journal Club, and always thought about these ideas as I was recording from synapses. And so I was very curious about how mechanical forces could be translated from really the level of a phospholipid bilayer. I was a phospholipid chemist before I started grad school for a while and synthesized all kinds of very odd phospholipids, but many that were biologically active, and having an understanding of phospholipids and how they exist in different phases, and kind of some of the physical chemistry of phospholipids, this is a very interesting idea that, mechanical forces completely could play a role in active ongoing signaling in the nervous system. And it was something that was overlooked. So most of my PhD was spent on recording from synapses and then going back and doing electron microscopy and confocal microscopy and very early multiphoton microscopy, and then in optogenetics. And there was this form of synaptic transmission when you remove all calcium, even when you have botulinum toxin and tetrodotoxin, you clamp activity, you still see ongoing synaptic transmission. And I was always very curious that if spines are moving and they adhere to each other through cadherins or extracellular proteins, could could the movement from the postsynaptic compartment be translated into functional synaptic transmitter release or neurotransmitter release from the presynaptic compartment, right? So kind of a rippling effect of the active zone, if you will. And I was like, that is really a fascinating idea, right? And my postdoc advisor, I went and started doing a postdoc and got into olfaction and some early optogenetics, synaptofluorin. And you know, it was like, this is a great idea. He's like, you know, well, no one's really going to care. And I was like, okay, well, that's probably true. So I moved to Arizona State University and started my first lab there, and really set out to develop an approach to study the mechanical forces of, you know, how mechanical forces, micromechanical forces affected neurons and synapses. That was the one of the main goals in the laboratory, is to start to pursue this idea and incubate it and see what would happen. And so we knew that from the Fry brothers, kind of a classic paper, that you could use high intensities of ultrasound to non-invasively modulate ongoing brain activity or sensory evoked brain activity. If you flash a light, you can see you suppress cortical action potentials. And so this was using long duration continuous wave ultrasound, and, you know, coming from synaptic transmission and plasticity and understanding channel kinetics and channel dynamics and the time scales, microsecond time scales and millisecond time scales of action potentials. You know, we decided to take an approach that would be non-thermal and use ultrasound in non-thermal mode by pulsing it, right? And keeping very, you know, different durations of pulses, but trying to keep it in a regime that we wouldn't heat the tissue or cause any macroscopic heating. And that was the basic approach. So we used ultrasound, and you're familiar with ultrasound. I don't have to belabor this point, but it's a mechanical wave. It induces mechanical energy and can cause local translation of particles without any net displacement, depending on the peak pressure and negative pressure. It has some interesting advantages in tissue, and I think that, you know, these are things that you probably discussed, that you can more precisely target tissue compared to electric fields that are you can't really control electric fields in tissue. It's very hard a priori to control them. And you can perhaps a little bit better do that with mechanical waves, and that still remains to be determined, but I'll talk more about that a little bit later. So what we did is we built you know, many of you may have seen this in some laboratories that you work in. This is an old microscope. We liked it because you could modify it pretty easily, and Olympus wasn't opposed to that. We didn't have to do any modifications for this particular set of experiments, but we built a rig that was designed to investigate whether ultrasound would affect synaptic transmission and ion channel activity. And so we had an ultrasound transducer, and then, you know, basically a water dipping lens, and we used organotypic slice cultures. You see this Schneering image representing kind of the ultrasound propagating up through the tissue. And we used organic calcium indicators and organic sodium indicators to image sodium and calcium transients, and we showed that we could stimulate action potentials as well as sodium and calcium transients. This was blocked with the addition of tetrodotoxin, indicating that they were voltage-gated sodium channels. We also showed, we used synaptofluorin mice. When I was a postdoc at Harvard, we showed in Vinky Murthy's lab, we made the first synaptofluorin mice, and we used synaptofluorin mouse tissue to create organotypic slice cultures, and showed that we could also affect synaptic transmission or synaptic vesicle exo and endocytosis. We then got a phone call, got an email actually, probably about 6 months after that paper got published, from Bill Newsom. And I was like, you know, a lot was going on. We started to like get a little bit of traction. I got an email from Bill Newsom. I was like, hey, this is cool. And like, Bill Newsom emailed me, like somebody actually knows from the neuroscience world, knows I'm alive, right? Because Bill Newsom's he's a powerhouse in neuroscience. And he was like, this is a really fascinating idea. Could you use ultrasound to generate a phosphine in the visual fields of a non-human primate? And I was like, you know, I didn't know that much about ultrasound and how to use phased arrays, but I was like, you know, I can make a spot and try to shoot a spot into the head, and like, you know, kind of like this, I don't know, maybe Alabama engineering, but it works. We created these acoustic collimators, these tubes, because the transducers were large. We were getting transducers that were built for non-destructive testing, and they were like 2 centimeters in diameter, maybe a little bit more than that in some cases. And so we built these very simple acoustic collimators just to try to get ultrasound energy into the brain to see if anything would happen. And we were able to do that. And then that's when I got to meet Kim, actually, because Kim and Randy King And Bill, you came over to ASU, and we had a day, we hung out, we did some experiments, and that kind of really started things, I think, in terms of, it's interesting if you can get something to work in slices, but I think it becomes less, if you can't get it to work in an animal really quickly, it's like, well, what's going to happen? And then, there are lots of like, well, so what about this? Did you check that? And it really started the field, I think, and it reinvigorated the field, these experiments did, because they generated a lot of questions.
Yeah, I just want to interject. Jamie was so gracious and hosted Randy and Bill and I, and we came down and Jamie showed us how it all worked in animals. And so thank you, Jamie, for that.
Yeah, no, it was great. It was great. It was great. It really did. Because for me, it was like, I didn't know, like I had known that some stuff, but I didn't know people were trying to do this, right? Because I was just trying to say, look, could you use mechanical waves, or do mechanical waves affect, transmission? It's an interesting question, and one that's kind of was forgotten by biology, to be honest. So yeah, I was happy to get some attention, and that was fun. So we showed, like we did as careful as we could, probably not at the limits of, not at thermal limits, but pretty good with simple devices that we could modulate brain activity without altering brain temperature. And so that kind of confirmed that there's a mechanical effect. But what is the mechanical effect? Like, what are the, like, okay, so we know that we can modulate voltage gated sodium channels, but what happens, what underlies the ability to modulate sodium channels is what's not known, right? And what was not known. And I think it's still kind of unresolved, but we've shed some light on it since then. The field has. So I'm going to start with this diagram. I got to write a paper in Nature Reviews 8 years ago now. It's crazy, it was that long ago. But kind of expressing some of these ideas about the brain is a non-Newtonian fluid. It's a viscoelastic material. The brain circuits are mechanically sensitive, and within the cells and even on the outside of the cells, most of the proteins both generate and sense forces, right? And so that's something that was a pretty mature idea in the world of mechanobiology, and looking at kind of like the viscoelasticity of red blood cells, for example, and studying fluid dynamics and problems of how red blood cells get through capillaries. And people had pretty mature understanding of mechanobiology. And so I was like, we need to integrate those ideas and those concepts with neuroscience, right? Because we're missing something. And so I got to write this paper. But what I want to point out here is that there's several points where ultrasound could be acting, where the forces of ultrasound could be acting to cause these effects that end up being observed in channels. And one is the membrane, right? So the phospholipid membrane, certainly a viscoelastic fluid capable of propagating mechanical waves in multiple directions. Actin filaments, right? So actin polymerization, depolymerization generates piconewton forces in the cell constantly, gives the cell kind of rigidity. Same thing with microtubules in terms of trafficking and moving things around cell division. So many ion channels are also known to be mechanically sensitive, and I think that's another point that it needs to be understood. But really, it's a composite, right? So all these, they're all anchored to each other. The membrane's anchored to the proteins, anchored to each one of the compartments, and so it's hard to push on any one of those nodes, if you will, and not affect a downstream, at least with some distance, right? Not affect a downstream process. So we started, I don't know if you remember this, Cam, but this was we really wanted to understand at a very detailed level what was happening. And so we started, I guess naively, I was like, I put a couple students, I was like, molecular dynamic simulations, let's do some modeling with some supercomputers. And so we got some time at ASU, and we started using non-equilibrium molecular dynamic simulations, and created this boundary condition that had a phospholipid monolayer, and then two phospholipid monolayers to make a bilayer, and then the other end of the box, boundary box, was another monolayer. And that was, we hydrated it with like 24,000 water molecules and sodium ions and chloride ions. And we didn't have enough, at that time, there wasn't enough computational power. There was, but it would have taken a month to run one simulation. And the way these worked is like, you run a simulation, you go tweak some parameters, you run another simulation. And so we try to keep the model simple, but we couldn't model very long time periods of ultrasound, right? So what we did is we modeled ultrasound as an impulse wave. So just the front of the wave, if you will, hitting the cell membrane. And so what we saw is there was a redistribution of the phospholipid bilayer as well as ions in relation to the phospholipid bilayer. And that was, you know, like, we didn't go on and publish it. We had a collaboration that we started with some folks in Japan, and I think it was very interesting, but the models were really hard at that time. And I think people should start to return to these models. The computational power has it's massively increased since then. And they're very powerful models if you have people to run them. We then took those ideas and went to Matthias Schneider's lab. He was at Boston University at the time. He's a mechanical engineer, but he's very interested in non-electrical action potentials and how mechanical action potentials propagate in nervous systems. And so we know that, like, when you stimulate an action potential, there's actually a mechanical impulse that travels down the axon. And Matthias was interested in studying this and studying kind of the molecular biophysics of what happens at the membrane level during an action potential in a membrane without channels. So I went to his lab and we set up an experiment in a Landmeyer trough, and we used two different forms of phospholipids, multiple phospholipids, but two that were doped with fluorophores. One was a green shifted fluorophore and another was a red shifted fluorophore. And then we could use a FRET technique. So FRET is forced or resonance energy transfer. So if the green fluorophore was close enough to the red energy transfer, the photon would be absorbed by the red fluorophore, and then we would see a change in the emission in that band. That's just a short story of FRET. So you're actually measuring distance, right? The distance of the donor receptor pair. That's really the metric. And when we applied ultrasound to phospholipid monolayers, we showed that we could increase the surface tension, or we could modulate the stress-strain relationship of the monolayer by introducing ultrasound, which was probably then propagated some shear wave through the monolayer. So Matthias has gone on to study this. We think it's also another way to be very quantitative about how, like, what's happening at the level of the membrane. It's very hard to extrapolate what would happen from these experiments into an in vivo system, but nonetheless, it does shed light on kind of the mechanical sensitivity of the membrane itself. Along those lines, other people have talked about, you know, Shai Shoham has talked about capacitive effects that could be generated in tissue by ultrasound. And this is a paper that came out recently. It was last year now, but they showed in tendons actually that they could generate piezoelectric effects using focused ultrasound. And so they had to use a tomographic technique to be able to image the electric fields that were generated, but they could generate, you know, electric fields that were up to 80 nanovolts. Once they used corrected imaging methods, they showed up to 80 nanovolts in tendon. And so that's interesting, right? I mean, the idea that basically, if you think of the membrane as a capacitor, and you have basically these charged plates, and you're separating the distance from the charged plates, right? Like that's a powerful way to generate an electric field or modulate an electric field. And that could be happening in a microscale in the membrane. And I think it's something we shouldn't rule out. Another technique, so if you look at the cytoskeleton, this is also something we didn't, a lot of the work we did, we just didn't publish it. We wanted to verify, like intellectually and ideologically, do these things line up with the hypothesis, but keep our eye on the translational goal at the end. But this is work we spent some time on, and we were fortunate to get these actinin stress sensitive FRET proteins that it's an actinin molecule. And again, it uses a FRET technique. It's a different donor receptor pair, but the principle, the cerulean and venous, act very similar to the green and rhodamine, NBD and rhodamine fluorophores I showed earlier. So you're effectively measuring the distance between the donor and acceptor, and this is now on actin, right? So it's basically within actin, the actin filaments, you're measuring the stress in the actin filaments. And we applied pulsed ultrasound to the cells. We showed that you could reversibly, again, modulate the stress-strain relationship in the actin filaments themselves. So now you're thinking, we've seen this in the membrane, we've seen this in the actin microfilaments. The next obvious thing to look at would be ion channels, right? And so it's kind of the final tethering point. We had worked for a while on doing some recordings on channels. One of the channels that Jan Kubanek had shown here, actually, the TREC1 channel, we had a lot of work showing that we could modulate TREC1 channels and oocytes and single cell recordings in membranes. But Juan published this before we could get it out. And what was really nice here is he showed that you could modulate TREC1 channels, which I don't know if you know this, but TREC1 channels are potassium channels that are essentially like potassium leak channels responsible for maintaining resting membrane potentials in cells, as well as other functions. And then sodium NAV 1.5 subunit channels. These channels are one of the channels that are involved in generating action potentials. And so here we have an effect of using ultrasound showing that you modulate two very important channels and just maintaining ongoing activity patterns in neurons. Mikhail Shapiro's group has done a lot of really nice work, and I think they're really ahead of the field in the way they're approaching the problems and looking at mechanistically what's happening. But they published a paper recently showing that there's essentially signal amplification from calcium channels and sodium channels to calcium internal stores, right? And that's just naturally the way that neurons work. But it's nice to know that you have these, you can go in and dissect these cellular signaling mechanisms in great detail and start to find out where the sources of ions are coming from. And so I think he's doing a lot of work here, as well as I think a lot of work he's doing on molecular genetic techniques and being able to image. He's doing a lot of great work. So I look forward. I think he's going to do a lot for the field in terms of mechanisms. Piezo1 is a natively mechanically sensitive channel, right? So there's channels that are polymodal. Some of them, like trip channels, for example, will respond to pH or electric fields, or they'll respond to temperature or pressure, right? And so piezo1 responds to pressure. It's a channel that's known to be involved in somatosensation and many other, some other basic functions in cells. And this paper was really nice. They showed that they could modulate the activity of piezo1 using ultrasound, but not only did they get the piezo1 activated, they showed, similar to what Mikhail had shown, that you get downstream effects. And here the downstream effects are on intermediate early genes such as CFOS, phospho-CREB, phosphocam kinase 2, right? And so I think that's again showing that you get effects at the membrane, and you get these downstream effects. You're not blowing up the cell, right? So you're not activating the cell. You get these ionic currents, and there's something deleterious to the cell, and it's undergoing apoptosis or something like that. The cell's alive and it's still signaling and functioning as normally expected. So astrocytes are often forgotten. This is astrocytes are actually, when you look at it, they're more mechanically sensitive than neurons actually. And so I think it's interesting to think about how they may be playing a role. But Justin Lee's group published a paper late last year showing that ultrasound could modulate TRPA1 channels in astrocytes. And then that affected a BEST1 mediated release of glutamate that activated NMDA receptors, right? And so when we think about neurons and as a functional unit in the brain, we typically think of a neuron, an astrocyte, and then a blood vessel or a capillary, right? And so it's kind of the triad. And so I think it's going to be important to monitor astrocytes in the future. Now, so we've talked about channels and how you can modulate channels, and now I think that I want to shift a little bit to sonogenetics, and this is an idea that has been borrowed from many of the X-genetic efforts, but I would say largely borrowed from the most successful one in optogenetics. And so here, instead of using a light-sensitive protein, they're using mechanically sensitive proteins. And so they used a protein that was a TRP4 channel that was natively expressed in neurons, but they expressed it in a neuron that didn't normally express it, right? So it's a native channel to C. elegans, but they then overexpressed it in a cell that didn't have it. And what they showed is they could basically induce a behavior by activating that channel. And they've gone on now, they have other papers that they've been working on, and they've gone on to, this is actually using ultrasound to stimulate. This is a mimosa plant. When you touch the mimosa plant, it activates ion channels and causes the plant to close. And they activate, you can't see it, but there's an ultrasound transducer on this plate, and they're actually activating the ion channels in the plant using pulsed ultrasound, right? And so what they're doing now is they took this channel and they've cloned this channel and they're putting it into neurons. And now they're going to use, so they're going through just like a lot of people, I don't know how many people know this, but like when GFP was first discovered, right, it wasn't a very efficient protein. It didn't yield many photons. And so they had to go in and mutate GFP, and they ended up with enhanced GFP and titrate it for pH and activity. The same thing with optogenetics, right? Like as we started with, you start with green fluorescent protein and you end up with all these different colors and proteins. So they're doing the exact same thing with this now in many animals. And I think this is like kind of the you know, this is an interesting idea and they're pushing it forward. But then when you get to this paper, I think it's like, okay, it's coming, it's going to be here. So what they did is they took this protein Preston, which is an ion channel protein that's expressed in the auditory system and is responsible for helping with hearing, right? And so they took Preston and they overexpressed it in deep brain nuclei in the brain in areas where it wasn't expressed. And so people are getting really smart about this. They're going through and they're cloning, they're looking at species, right? And they're saying like, what are the channel properties? What could be the mechanosensitive regions of these proteins? And then we're going to put those mechanosensitive regions of the proteins and make new channels and generate channels and put them in different regions of the brain. And when you do that, it makes that region of the brain even more sensitive to focused ultrasound, right? And so you have very high selectivity and also high specificity. You know that you're going to activate those cells that have that channel because you've rendered them extremely mechanically sensitive. Right. And so this is, I think it's a fascinating field to look forward to.
Would you say that this field is like trying to compete with optogenetics in a way? Because, you know, it's obviously harder to deliver ultrasound than a light. And I'm open to it.
Yeah, optogenetics can't touch this. Right. So, and this is why. So you always need a form of light to get in the body. Right. And so this is one. You cause the mutation, the genetic mutation, and you use an ultrasound from anywhere and not have to worry about getting light into the body. So, and then on top of that, there's another paper, and I should have put this paper in, but they actually used ultrasound to pump photons out of a molecule that was expressed in the body, right? And so you basically then take a genetic approach and you express a protein in the body, and then you use the ultrasound to make those molecules generate photons endogenously. Right. And so now you don't need to put a fiber optic or anything like that inside the head of a mouse or a person. Right. So you say, look, imagine like if you could have a mechanically sensitive bioluminescence protein, right? And then say, look, we're now going to generate endogenous CFP, right? Or blue photons, cyan photons, and activate, you know, some endogenous optogenetic protein, whatever flavor you want. I think that's fascinating. So I think they each have their own place, right? But if I had to pick one, I'm going to bet on optogenetics. I mean, even before Carl got
to go even further.
Yeah.
So I was going to say, to go even further, you know, and I think there's a debate to be had about, you know, what will win in the future, and I'm up for that. But I think sonogenetics, if anything, if you thought about human implications, it's much better because the deep brain targets in the human are very deep. You know, there are new options now where you can actually transcranially illuminate them, and that's great. But for a human, do you see this ever ending up in humans, or is that so far away? Or
no, because we can control. I mean, it's hard to control. Controlling scattered photons is hard, right? People have spent, like the physicists have spent hundreds of years trying to do that, right? And we've recently had some success, but it's still difficult. But I think being able to control the mechanical energy is, because controlling photons is random in some sense, right? But controlling the mechanical ways and mechanical energy, I think you have a little bit more precision over that in the tissue, right? And so like just dealing with scattering, right? Like it's going to be interesting. I would be a fan of like and this is what the paper was, a sono-optogenetic approach, right? Because I think optogenetics is very, very powerful, right? And then the question is how specific like if there's a lot of if all these ion channels end up being mechanically sensitive, even if you overexpress a protein, you still have all the other neurons around that have endogenous mechanosensitivity that you could also be affecting. Whereas with optogenetics, only the photons are going to activate those proteins, right? So if that makes sense. So it could be that the endogenous mechanosensitivity causes it. I don't think it's going to be magnetogenetics, that's for sure.
Yeah, I know there's contested works after the original publication, but it's really good to hear your perspective. I think we look forward to you being maybe down the future one of the, you know, if you work with a company or build one for actually engineering this for a human safe version, if virus production gets there.
You know, I always had nightmares. I used to, because when I first started this, you know, I had people, I was like, well, what could you do with this? And I was like, look, if there's ever a robot that like flies and like injects you in the eye with some sonogenetic protein, I called it acoustogenetics back in the day. We used TRPC channels to show that it would work. That would be scary, right? Like it's like basically a drone flies up, and then it's basically you're rendered mechanically sensitive, you know. So it's going to be interesting. I think optogenetics is like, it's got the advantage it has is the field, right? Like it's massively studied and we understand how it works, right? Like we still are like, this is mechanobiology in the brain is a very, very new idea. So this is where I think actually we'll have some of the biggest breakthroughs. And this is a paper, I don't know how many people notice this in the field of ultrasonic neuromodulation, but I was really excited when I read this paper. So what they did is they used basically MEMS actuators and combined it with atomic force microscopy and these cantilevers, and they could go in on these microposts and actually touch neurons, right, with like high amplitude force or low amplitude force, and monitor responses. And so it allowed them to be very quantitative. They're trying to understand where on the neuron, like there's basically the most mechanically sensitive of the neuron, right? And so I think these types of approaches, when you look at what they've been able to do, so here's an example of the experiment just so I can show you. I'll play this movie, hopefully it will work. You see this cantilever comes down and touches this neuron, right? And you activate a calcium transient. And because they know they can quantify the force, right, then they can create these maps, which is really freaking cool, right? So like you create this map and you can look at like in the soma, the blue and the green and the purple is interesting because so with low force, most of the cells showed a transient response. So you see this transient response. And then when they had a higher force, fewer of the cells showed a transient response and more of them showed a sustained response, right? And then they characterized the null responses. And then when you look at the map, they actually show on the map, right? Like you can see the green responses of the transient and the purple. And there's like saying kind of what I was saying earlier, right? Like tubulin, actin, these are all potential sites in the membrane, and you can even see with ion channels, right? So being able to quantitatively characterize like and map individual neurons and force like this and how it affects it, like it's really fascinating, really fascinating. So I think that this is like, this was edited by someone at Stanford. I don't remember who, but this is, it's going to be, you know, this is a very, we can't do this with ultrasound, right? We can't quantify the force at this level with ultrasound using the techniques we have right now. And so using atomic force microscopy should be okay. Can you hear my daughter crying? Okay, we're good. Spatial resolution and targeting. There's going to be a lot of advances in this area. I think right now we already know that transcranial focused ultrasound has the highest spatial resolution of all the non-invasive brain stimulation method techniques, and it's just going to get better. I think there's room for improvement. There's a lot of barriers we have to overcome in understanding how to control and deal with aberrations induced by the skull, which I'm sure you've talked about. But those things can be overcome, and I'll show you some examples of that in a minute. What I want to illustrate here, yeah, sure.
Could I ask you just to go back for a second to what the thought you had on the last slide, which is here you're sort of differentiating between different parts of the neuron, but there's also the thought that, you know, different pulsing parameters will activate different types of neurons or different ion channels. What is your thought about that? Because I'm not sort of clear exactly, you know, like what's the effect of different pulsing parameters? Were you going to talk about this later? I didn't mean to kind of jump in.
No, I wasn't, but I think that different pulsing parameters, I think it comes down to bandwidth, Kim. And I've always said this, right? I've always said that you need broad bandwidth until you understand the transfer function from mechanical energy to cellular activity, and many of them, right? Like I think that's going to be very difficult to then say this particular set of pulses works in this way. We use very general rules. We actually believe that similar to this paper, that low intensity and shorter pulses are better for exciting neurons, and longer pulses with more moderate intensities tend to be more inhibitory, right? And whether that's true or not, I don't know, right? And I think it remains to be seen. We always like the pulsing parameters because it gives you bandwidth, and then you're trying to modulate, like you're trying to identify with the ultrasound. Think of the ultrasound as almost a search stimulus, right? You're trying to identify the proteins that would be responsive to those frequencies and those timescales. And there's mechanically sensitive proteins and neurons that respond on the orders of seconds, and there's mechanically sensitive proteins that respond on the order of microseconds. Right. And so that's why the bandwidth always for us seemed to be really important. And so the more bandwidth you can introduce, right, the better off you are.
When you say more bandwidth, you just mean like higher frequency?
No, multiple frequencies, right? So multiple frequency components. So this gets to the idea we always had about low Q versus high Q transducers, right? Like we're always like, look, the broader band transducers are going to be more effective at the end of the day. And if you use a pure tone, you're less likely to affect activity.
Okay.
And then you can basically, because of the intensity, you can start to, if you have a pure tone, right, you can basically build up intensity really quickly and generate thermal effects. So lower intensity spread across the spectrum in our hands has always seemed to work better.
So that's sort of interesting, because I think we've been thinking kind of going in a direction that's sort of counter to that because of wanting to get rid of the auditory confound. So thinking
Everyone always has.
Or,
Everyone always has. That's it's been a point of contention between me and everyone else in the field. Everyone is always like, oh, but I think they've started to realize it, right? And even Stores Medical mentioned it the other day when they use a shock wave, right? It's a 1 microsecond shock wave, but they're like, that 1 microsecond shock wave is a broadband ultrasound stimulus, right? From 20 kilohertz up to like 20 megahertz. And so, and again, I don't, like in the future, if you had a cell and you could, like, if you had high frequency ultrasound and could really control it, I do think different pulses, different pulse shapes would give you different effects in different regions of that cell. And that's what this is showing, right? This is showing that different pressure profiles are giving you different effects in the region of the cell. You should be able to reproduce these pressures using ultrasound to some degree.
okay, that's really interesting.
Right, because if you think about, like, I don't know what it would be, like maybe 10 cycles at a megahertz or something when this, as soon as this cantilever, the first touch, when that cantilever touches, I don't know what the pressure is, like it's 5.8 kilopascals, right? That's actually pretty high compared to what people use for ultrasound. So it's interesting. I just think that this method, right, it's just powerful in terms of understanding, again, because we don't understand the basic principles in terms of how the brain's mechanically sensitive, right? We're trying to make it fit an electrical organ. We understand Goldman, Hodgkin, and Katz, and neurophysiological excitability, cellular excitability, but we have no idea how those things relate to kind of the mechanical biophysics of cells. Right, it's completely understudied in the brain. And what's cool, if you look at like magnetic resonance elastography, and you can look at like how there's different, and you can create an elastogram of the brain, right? And you look across disease states, you see changes in stiffness in the brain, just like you do with atherosclerosis in the heart, right? Like there's stiffening of the brain before you actually see some of these emergent properties of the disease. So there's a lot to uncover. So the spatial resolution now using 0.5 megahertz is on the order of the, so this is a beam that's what, 5 millimeters across in diameter. And you can look on this, and as you can see 5 millimeters for the spatial extent of the electric field generated by a deep brain stimulating electrode. So you're on the order of what you can do with a deep brain stimulating electrode in impaling the brain, right? Which is impressive. So where that can be improved, and this is another area that I got really excited about. I get excited about holograms too, like holograms and metamaterials, and this combines both. So what they do is they kind of created a metamaterial, if you will. It's a 3D printed holographic plate, right? Which is basically an arbitrary pattern that's 3D printed on a plastic. And I think this is really fascinating for, like, if you look at low cost, because the whole point of this paper was like, this is low cost method because this is a piece of 3D printed plastic that anyone can print at your house, and it's kind of a messy surface, right? And we're now using that messy surface as a lens, as a metamaterial, as an acoustic metamaterial lens to generate holograms in the brain, right? And so they showed that they could do this for transcranial focusing, right? So they could use arbitrary acoustic fields or arbitrary ultrasonic fields and then generate acoustic holograms from those. And I think that if you just look at the low cost method of printing these plastic plates essentially, and combining it with just a single element transducer for focusing, it could be a powerful method, right? It could be a really powerful method. I think computationally it's going to be intense, and it's going to require a lot of, there's going to be some caveats about the skull, and it could take, like, it may not be, you might be able to do this, but if it takes 4 hours to generate the hologram to then project into the brain, or 24 hours, right? then it's how good is it? And so I think that remains to be seen. This is another area that I think is interesting, and it's not something that I don't completely understand the mathematics behind it, but this group is taken images or imaging methods that were borrowed from seismic imaging, or using ultrasound for geophysical mining. So people look for, you can look in the Earth's crust using ultrasound to look for gas pockets, for example. And so they use this method, it's called full waveform inversion, which will basically go through and estimate the speed of sound or wave velocity in different media. So this is their ground truth. This is a brain and a phantom surrounded by ultrasound transducers. Here's 1,024 ultrasound transducers. These are all in silico experiments, by the way, right now. And they start with just water. So this is a water-filled phantom, and they train their model. And as they train their model, they basically can end up with a computed image that looks very similar to the ground truth, right? Using this full waveform inversion technique. So now what they did is they used the same method, and they wanted to see if it would work with a skull. So they introduced a skull into the model, and they pointed out that, and so over here, this is basically the speed of sound in the tissue, right? And what they did is they show here, they wanted to kind of make the lookup table so that you could actually see inhomogeneities in the skull, right, which you can clearly see. And those can again be computed by the full waveform imaging method using this 1,024 transducer array. So now this is what it looks like. It's kind of cool when you look at kind of going through and building up the stack from the ground truth, starting and reconstructed. This is one of the supplementary movies. It's actually really kind of cool to watch. This is now just going through the brain, and you can see the skull, right? And remember, this is a water-filled phantom because that's part of their learning. They start with that and they build up across the tissue, and you can see the transducers and some of the noise coming in here. But very nice, clear image of the skull with the 3 different layers, right? And this difference in the speed of sound in the 3 different layers of skull bone. And so again, this is something, I don't know how computationally intense this is. I was working for a while with some people from Los Alamos National Laboratories. The Department of Energy does lots of things related to this, and some of their guys are reverse timing algorithm people, and they worked on geophysical applications. And I know they started to work on methods that are very similar to this. If you could do this, then what that does is it eliminates the need to send the patient to have a CT scan to calculate the bone density to be able then to accurately project ultrasound across the brain, and you get a pretty decent image of the brain on top of that too, right? You don't get the functional aspects, but I think if you look at some of the work that Mikhail Tanter's been pushing for years in terms of his functional transcranial Doppler methods, if you have something like that and you can get to those, like if you can combine these types of methods and those types of methods in terms of signal processing and dealing with noise, who knows, right? And that's why I think ultrasound is really exciting, just because it's powerful. It's gonna be really powerful. So this is the last image. They actually went on to show that they could model or image a hemorrhage in a brain. They wanted to apply this for stroke, so basically to be able to image someone really quickly and identify a stroke volume. But what they did here is they used just an acoustic test tank, a skull, and they show single source with a receiver array and just their signal characteristics. So I think it's an interesting paper for the people that can understand these processes. It's a lot of signal processing. Therapies and treatments. So we've sponsored some studies and worked in collaboration with John Allen's laboratory at the University of Arizona for many years now. This is one of the first devices we made, and one of the companies that I had. And we this is a battery powered device. It is programmable from 0.5 to 1 megahertz. It had a built-in sham mode. It was pretty cool. It was all blinded. You had to have a code to get into it and run it. So we did a double-blinded sham controlled study, 2 studies, and we showed that we could modulate default mode network activity as well as mood in depressed individuals using this device. So we are going to continue now in my academic laboratory. That work is then that's going to, the push on depression and mental health disorders will take shape through the company. In the academic laboratory, we continue to study, we're really interested in emotional attention and kind of like awareness, emotional awareness. And so we've been doing So how your brain processes emotional mistakes, right? Like, it really impacts mood and mental health in ways that people don't understand. And that's what the default mode network is related to, right? So like, it's kind of like yourself and your person, where you fit into the world, and making mistakes in those networks is what causes people to have these, kind of negative ruminations and bad thoughts. And so we're trying to understand if we can move that type of behavior around. So this is work my PhD student, Maria Finney, she just finished, my last PhD student. She just finished last week, but she built this pretty elaborate rig. She sewed this head net to keep the transducer in place. It's all neuronavigated, 32 to 64 channel EEG. We have used headphones, so we record the sound of the transducer, and we use the sound of the transducer in our sham experiments. So basically, the subjects, all they can hear is the same tone, regardless of the condition. Everything else is fixed the same, right? And so they're isolated, right? And they look at this monitor. The experiments are pretty cool. We have it set up so that there's a psychophysical test box. And she used in this task a modified flanker task, which is a psychophysical test. Like, you monitor these arrows and you indicate on the keypad whether the one in the middle is pointing to the left or the right, right? So you just look at this one in the middle and it's like, is it left or right? And so what she did is she put an emotional distractor in the background. So she put these different faces, that like fearful faces and scary faces, and those are in the background. And so your brain's trying to attend to the task, but you have this emotional distractor in the background. And we know the neurobiology is beyond the course of the discussion of the class, but we know that the dorsal anterior cingulate nucleus and the insula are both implicated in these types of behaviors. And so she had two papers. We're going to hopefully publish them back to back. One looking at the dorsal ACC and one looking at the insula, all neuronavigated, every subject had MRI. And so what we found is that you do modulate the error potential, right? And so the error potential is shown here. So you modulate this error potential. When your brain knows you make an error before you know you make an error, right? And you will generate an error potential, kind of like, go back and look at that because we just made a mistake, right? Slow down, take a break, and that your brain does this automatically. And it's part of the default mode network. I think that's one of the coolest aspects of the study. We found that you can modulate neurophysiology and EEG and widespread circuit activity, as other people have reported with transcranial focused ultrasound. The point that I want to point out about this set of experiments is one of the things we're trying to do with the NeuroFuss unit we're commercializing now is we want all this to be turnkey for investigators, right? So because it's hard enough to build the ultrasound equipment and then set up the EEG. And so the partnerships that we've developed, and the reason we've developed them is because I wanted this to be turnkey so that people don't have to worry about things they shouldn't be worried about. They should worry about their experimental design. We'll make sure everything talks to everything else that you need it to talk to. And so that's kind of the point. And I'll come back to that in a second. A couple of the last points I want to make is—
Hey, Jamie?
So, yeah, sure.
Oh, yeah, before you go on, you were saying that you played a tone in order to make sure that you had an appropriate sham. How did you know what tone to play? How did you know what their perception was?
We record the transducer. Yeah, so we record the transducer, and that's what they hear either way.
Oh,
so and then we ask them. So we ask them. It's pretty loud too. That's the other thing, is we turn up the volume, and so we ask them if they can't hear the transducer at all. All they hear is the tone that's played through the headphones.
So what you're asking is the tone of the transducer itself, or turning on the equipment, not what kind of the PRF effect on—
No, that's what it is. So they couldn't detect the PRF on the skull, because that's played at the same time the sound is played, and the sound, I think, masked whatever the PRF sound is generating. Because it's a really loud sound. I should have had a sound of it, but we recorded the transducer with a microphone. It was bzz, bzz, and that is a loud sound that's played at the same time that the transducer goes off. Right. So that's like the best that we could do. And, I think this issue about sound, it's interesting, right? Like, people forget transcranial magnetic stimulation is really loud. When you send several hundred volts into those wires and that coil, right? It's loud. It's click, click, click. And that clicking has actually been shown to cause plasticity via the rubospinal system, right? The red nucleus. And so the same thing with TDCS. I do a lot of work on auricular vagus nerve stimulation. We spent several years for the Department of Defense working on designing the method so that it was imperceptible to individuals, right? So that was a real art to be able to get vagus nerve stimulation, electrical currents to go into the ear such that someone couldn't even know that it was on or not, but at a sufficient level that would do something biologically, right? And so this is just a non-invasive neuromodulation, non-invasive brain stimulation, something we've been dealing with forever, right? And I think there's no surprises. You just have to design the experiments the best that you can. So what's interesting is GE is now in the field, right? So this is work that was sponsored by the Department of Defense, by DARPA, as well as internal work at GE. So speaking of vagus nerve stimulation, they have shown, so vagus nerve stimulation, when you stimulate the vagus nerve in the neck, cervical vagus nerve stimulation with an implanted coil, it causes bradycardia and can cause other cardiac side effects, right? You have baroreceptors that are right there. It's, you know, it can cause like muscle contractions in the neck. Because of those cardiac side effects, that world of bioelectronics, using vagus nerve stimulation to modulate immune function, they've started to use now ultrasound, and have shown in multiple papers that they can use ultrasound to modulate vagus nerve activity as effective as electrical stimulation without the cardiac side effects, right? And so in this particular paper, what they showed, they're very excited about this, by the way, and I am too. What they showed is they could use focused ultrasound and activate neural activity in nerves that innervate the spleen. And that resulted in kind of activation of classic cholinergic immune pathways and reduce cytokine activity, right? When they do the same thing and modulate hepatic branches of the vagus to innervate the liver, they could then modulate glucose metabolism. And they did this in DB mice and OB mice, and they showed that like the goal is like now you can treat diabetes or obesity if you target the liver with focused ultrasound, and if you target the spleen, you can then activate these kind of classic cholinergic immune pathways. And so Medtronic is not going to let GE do this by themselves, of course, right? So Medtronic also had a grant from DARPA and the Department of Defense in collaboration with their colleagues at the University of Minnesota, Hubert Lim et al. And what they showed here is that, again, you can modulate the activity of the splenic nerve, innervating the spleen, and activate these anti-inflammatory pathways, right? And so decrease the level of inflammatory cytokines. And they showed that they could treat inflammation in animal model of rheumatoid arthritis. And my understanding is they're running human trials on this now, using ultrasound transducers that were bought, for lack of a better term, off Amazon, and scanning the spleen, seeing if they can treat rheumatoid arthritis, because why not, right? So doesn't have imaging, but whatever. I think it's great. I think it's a great story. It shows this level of industry investment and involvement, showing that they can start to drive things therapeutically. It's just going to move the whole field forward, right? There's now even more questions that we have to ask, because now in the periphery, you have kind of the same thing that's happening in the brain, and the peripheral nervous system is just as powerful as the brain. Make no mistake about it. So hardware, really quickly, I'm going to skip the left-hand side. One of the purposes of this paper was to just verify from a neurophysiological perspective. Leonid Gavrilov had done a lot of work in the 70s on using ultrasound to induce somatosensations, right, and auditory sensations. And so we wanted to come back and look at his work and just say, look, if you put a modern neurophysiological signature on those types of experiments, do you see something that the brain would make sense of, right? So does it seem to be kind of a natural phenomenon? And the answer was yes. It looked to be no different than a mechanical vibration on the fingertip or anything else in terms of brain processing. What's interesting about that is, look, Facebook has, talked about they want people to hear with their skin, right? And so one way to do that is to use ultrasound transducers on the surface of the skin, right? And so to encode vibrotactile information, almost like haptics, right? And speaking of haptics, you can actually use ultrasound transmitted through the air, and with radiation force activate somatosensory receptors in the skin, and generate acoustic holograms, right? And there's a company that does this, Ultra Haptics. And what's really cool, going back to holograms, is they have started to, this is just an artistic rendition. This is a collaboration they have with Bosch and some other car companies and home appliance things. I think they're actually using it in slot machines. But what it does is it projects a optical hologram onto an acoustic hologram, and then an object that's not there, you can reach out, see it, and touch it, move it, turn the volume. So that's a very cool method in terms of interactive virtual reality, virtual immersion type of world, if that's the type of thing you're interested in. I think MIMS transducers, MIMS ultrasound transducers, we talk a lot about CMUTs and PMUTs, but I think people forget that there's other methods of making transducers, and even using traditional PZT materials on flexible PCB boards will allow you to make wearables. And so building very small microscopic transducers that can be implanted in the body or built into fabrics or helmets or other medical devices, I think this is going to be, this is going to be a really fascinating world. It's cost intensive, right? Again, these are the types of things you spend a lot of money going through the fabrication of one or two pieces, and then it doesn't work and you got to go back, and it can be trying. But this is a method that they actually used. What I was just talking about is basically diced PZTs on a flexible PCB. And when they did that, they basically have a modulation array as well as an imaging array on this flexible PCB, so that they can image. This is the carotid artery, the jugular vein, and they're basically looking for the vagus nerve. You can see the vagus nerve. And so here, this is in the tibial nerve, and both of these have been shown to be modulated by focused ultrasound for different applications. But now you can image the nerve as well as see the nerve. And so if you wanted to have a wearable device that could, respond to you moving around and always track and image the nerve and keep the eye on the target, if you will, these are the types of devices to keep a lookout for. Kind of lastly, what we did with this is the world that I guess I came into, is like you have all these different pieces you kind of buy off the shelf, and you put parts together and you build a monster to generate this little tiny beam of ultrasound. The amplifier weighs like 40 pounds or something like that. It's ridiculous. So what I did, so my company IST, we've done some work for large multi-billion-dollar corporations recently, and I went to Sonic Concepts and I said, look, I want to basically work with you to create a brand, but the important thing about the brand is that we consolidate the field in terms of getting a handle on calibrated systems that everyone can rely on, and we know what they're doing, and we make it easy to use, user-friendly, and safe, right? And so Kyle Morrison and I went together and went and got BrainBox Limited. They were involved in very early transcranial magnetic stimulation work 20 years ago. And so we started working together. We've worked together for 18 months to create a turnkey system, and we've launched that. But I would just want to give you an idea of some of the things. So there's only 6 subscribers. We do have a YouTube page, but this will grow. But what we want to do is we want to create like we're just getting work. I mean, COVID hit, right? Like my company was supposed to be acquired, and COVID hit. That didn't work, and so we're like, okay, we're just going to stay the course on NeuroFuss, right? And we have, and I think it's worked out really well. So this will come up to speed very soon, but I encourage you to check it out. What I want to show you, and I think this is interesting, is so this is a video. I'll talk through it. So this is the unit, the NeuroFuss unit. This is a 4-element phased array, concentric array. We have arrays that, like, if you want a 1,000-channel array, we can make a 1,000-channel array for you. So the technology is there. I want to show you, it's a touchscreen interface. You can control each one of the 4 channels independently. The frequency, you control the focus on this little focus knob. So how cool is that? Real time, right? Okay, so then this shows this. So I want to show you this next one, because this is the piece that for me was one of the most important. I was like, look, this is what I want to see the field move forward. And so it's our SDK and our GUI. So just, I'll let you watch it. I'm going to talk about it. Very cutlery, brain box, really hard on this. This is the cool part. All the pages that populate that's it. That's what we're trying to do, right? I think that we want it to be as easy to use and for people to be reproducible, right? We sat through the Focus Ultrasound Foundation meeting this week, and we listened very closely to what the community is asking for, and we are committed to making sure that we deliver that. We think that the field has a lot of room to grow. We think that the field needs to be, like when you work with voltage and you measure voltage, you know what the voltage is, right? Like you can't even, two people can't agree on a measure of an ultrasound field by any means, right? So it's not like you have a voltmeter you can just hook up. And so those things have to come like full circle. And I think now what I've heard over the past, I guess 4 or 5 months is that it's time, right? Like we now know there's something happening, the field's maturing, we need the tools. And so science raised this issue recently in a nice article that I think highlighted a lot of work and controversy in the field, and they were cautiously optimistic. But they said that if we can address these issues and answer some of these questions, then we have a real tool that can do something and change the way that we approach interfacing with the brain. That's it. Thanks.
Hey, Jamie, we're going to have questions for you.
Sure.
Simple question here. The system you were just showing us, you said there was 4 channels. What do the 4 channels look like? Are they 4 annular rings or what?
Yeah.
Okay.
Yeah, they're 4 rings. It's 4
okay.
rings. But that array can be 16 channels, right? And so then we're basically 4 rings diced into 4 elements each, right? And then we've got a system that we're working on now that I think would compete with the Inside Tech system. That's a bold statement, but I think Kyle will have to come out with that when he wants. But I think we have a system it's pretty cool.
Yeah, I just wanted to let you know that one of the things that students may be very interested in is not just industrial opportunities, because that'll be interesting to them, but also how do you manage working in industry and in academics at the same time and balancing both of those? And so
for me, it's not a problem because my industry has driven everything in academics, right? I've been fortunate to be in a position where the university, wherever I've been, has embraced that concept and said that this is the way I'm going to operate. I haven't been able to get money from federal grant. From the Department of Defense, I've been really fortunate. I worked for DARPA for 10 years, right? And that has been an amazing experience. But as a investigator, you kind of want NIH money or NSF money to prove out your ideas. And I wasn't fortunate to be able to get money from those organizations, and so I had to find another way, right? And so for me, it's been the way. It's been the way, right? And in some sense, like in terms of conflicts of interest, right? Like it's pretty simple. I keep that. It's very parcellated and strict. There's no crossing of boundaries. I tend to make most of my students work on what they want to work on, and what they want to work on is not what I want to work on, right? So I have a smaller lab with fewer students that do what they want to do, and I support that as long as it fits in the kind of broader scheme of things.
Yeah. Hey, Keith, I know you're going to have questions.
Yeah, I had a couple. I thought that was great that you have this interface that allows you to select a paper. I think
Cool.
We were listening to Wynn and Leonard, like they were sitting there in the discussion the other day, and they're like, well, where'd you get your parameters? And they, it was a lost discussion, and to actually just have it load up is it's a neat idea. I think that's cool.
No, that's the thing we're most excited about. We're like, because it's so hard when you look at the parameters, you like,
oh my God, it's so confusing.
It's hard to figure it out, right? And I think that's one thing that the field needs is just nomenclature.
And in fact, we were just talking about that and starting up a working group on trying to inform people as to like what should they be reporting in their papers and things like that.
There should be a standard, right? Because it's all over the map. I mean, it really is. And when you go, like I had to build these tables. So like last year I built these tables with the papers, and I started going through the tables and like all the parameters. It's a wreck. It's a wreck, right? And I don't think that's anyone's fault. I think it's everyone's doing the best they can. There hasn't, all this stuff was defined for imaging. Right. And like what we do is a little bit, it is different than imaging, right? The pulses are longer. And so I think we have to kind of redefine things.
I had a historical question too. To go back to your original cell culture experiment, my understanding is you didn't expect to just see them directly activate. Maybe you were looking at a postsynaptic connection. Were you like, what, when it happened? Or did you think it would happen?
What I expected to happen happened the second time. The second time I clicked the mouse button to activate the transducer, what I expected to happen happened. And that was, I was this synaptofluorin organotypic slice culture. And I expected to see synaptofluorin signals. But what I thought was happening is not what was happening. Because what's in that paper, not many people talk about, is we basically blocked synaptic activity with botulinum toxin. We could still get synaptic transmission. I thought that you were basically driving synaptic vesicles into the active zone, and that would be the end of the story, right? Like it was just, I was chasing mechanically activated synaptic transmitter release, and like, oh, maybe ion channels, like it could all happen, but that's what I wanted. That I was like, this is the one thing I'm looking for. And it happened, and then all the other stuff happened, right? And I think it just became it was like, whoa, this is actually, there's a lot of cool stuff going on.
Right. So in that line, and like, let's just assume, we're all in the same boat here. I know there's some haters out there. They think it doesn't work quite like it does, but let's forget about that for a second. Why were people sleeping on this for so many years? It seems like such an obvious, good technique. And like, again, in my own hands and Kim's, like, it just works. Why did it take so long as to you to come back to show that it was going to be a great modern tool for neuroscience? And
I think it was the tools that we used to show that it worked. I think so people had shown that you could use ultrasound and modulate activity. So if you use ultrasound and you sonicate tissue for long durations and you stimulate an action potential, you can modulate the amplitude of the action potentials, right? And so that had been shown in hippocampal slices. What hadn't been shown was the direct stimulation. And I think what was, from our perspective, was we're coming at it differently, right? And so using the pulses and the short durations and the methods that we used, right? Like saying it's action potentials, it's voltage gated sodium channels. We spoke the language that all neuroscientists speak from a synaptic perspective. And the literature had never done that. No one in the literature came from a synaptic background. They all came from biomedical engineering. We're going to burn the brain, right? Like we can build this. It was clinical, right? You're like treating diseases without even knowing what's happening. And then some people started to get, they had some physiological, but you had to speak the language that neuroscientists understand, and that's synapses, ion channels, tetrodotoxin, voltage-gated sodium channels, voltage-gated calcium channels. And I think that's what allowed it to the neuroscientists then paid attention, right? Before that, the neuroscientists could care less. It's like, oh yeah, I can heat tissue, right? Whatever, I can do that with an infrared laser.
I don't want to ask too many more questions, but I mean, thank you so much for all of that. And I do imagine, and like it'll take a lot of work, especially from you and your efforts now, but I imagine like multi-billion-dollar industries across psychiatry sort of emerging from this.
Psychedelics. Come after psychedelics.
Yeah, sure. I mean, even in that direction.
Well, no, I want to combine. That's why I want to combine psychedelics with neuromodulation. I think that's the future.
Can you flesh that out a little bit? I haven't heard this. This wasn't in your talk, so.
Yeah, no. So I have a company in Canada that's we've been helping for quite some time on. It's a psychedelic company, drug development company. We have actually like very well-known professor emeritus, a couple of them, and drug development people working on psilocybin. And so I'm curious, like, one thing that we don't know about neuromodulation and focused ultrasound or transcranial magnetic stimulation is the dose response curve in terms of how often you have to dose people. Right now it's like 6 weeks for RTMS, right? So I'm curious, the world of microdosing and psilocybin and psychedelics, can that be, because they're the jury's out too, right? Like, how effective are they? Do they work? If you take that and combine that with another therapy that's, like you do once a week, that could be really interesting. Because compliance will be an issue. If someone has to come in every single day, we're nowhere near just giving people devices to go home and put them on their head that are transmitting ultrasound in their brain. That's just not going to happen, right? But having people come in once a week, is plausible. Every day, people just don't want, like, it's a pain, right? You got to go. So the question is, like, can I give you a gel cap of some really, really pure psilocybin and shoot some ultrasound in your head and make you feel great? The FDA will love it.
Yeah, it sounds interesting.
Well, it's no man's land, right? It's like, it's neuro, and when you think about, like, for me, the reason I got involved in the psilocybin company is, like, I started thinking about, like, consciousness and stuff. Like, that's a freaking, like, I'm in.
Yeah, you know what, we always say this to people, Jamie, like they can leave if they don't want to keep listening, but can you comment on, like, I mean, Jesus, is there a vast space of things to be done with ultrasound, or what? Like, you kind of opened up. God, I don't even have any comment. Do you have a comment on that? Like, it's much bigger than people know, and they're building these new weird tools in it, and it opens up even more space, and it's like, seems kind of endless.
Well, I mean, there's people doing like, so, you know, there's efforts that are involved in like spinal cord, like, because people are like, and I don't see this in the literature a lot, but people are working on spinal cord regeneration, peripheral nerve regeneration, the drug. I mean, I was always fascinated. Like, we used to use these cage molecules, right? Like cage glutamate, MNI glutamate. This stuff was so expensive, but doing that caged molecules for like like releasing, sonoacoustically driven materials from nanospheres, like that stuff is fascinating. It's fascinating, right? Because with other energy modalities, it just wasn't really feasible. Now with ultrasound, I think you can, especially with imaging. If you look at what people are doing with like point of care ultrasound and the devices that they can make, very small devices, you know, with CMUTs and AAC, it's like, whoa. That's why I tell the students, like, light's interesting, terahertz is really interesting, ultrasound is the most interesting. Right, like I'm fascinated by terahertz, but water exists, and it's like, if water wasn't in the body, like so many things you could do really fascinating with terahertz. But it's ultrasound. Kim knows too.
All right, Keith, do you have more questions?
I'll leave it at there.
Okay, well, Jamie, this has been amazing. You've really, you know, talked about a lot of things that really kind of open our eyes about new things to think about. So cool. Thanks so much. Really appreciate it.
Sure. I can't wait to come see you again, Kim, whenever we get out of COVID, whatever is happening.